This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 712, recorded on January 27, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's a bright, sunny day after a lot of snow yesterday, a medium amount of snow, and it's 21 Fahrenheit, which feels like 10 Fahrenheit. So that's -6 Celsius, feels like -11.
And from Austin, Texas, Rich Condit.
Howdy. Another beautiful day. Sunny, 54 Fahrenheit, headed for 65. After I hang up from you guys, I'm going to go spend the rest of the day in my yard.
Today we have three guests for your virology learning experience. They are all at the moment in Germany. And let me introduce them to you. First, from the University of Veterinary Medicine, Hanover, and Umeå University, which is in Sweden, Gisa Gerald. Welcome to TWiV.
Thanks a lot for having me. Hello, Vincent, and everybody else.
And Gisa, I met at a meeting in Sweden 2019, right?
Exactly. And that's why I was lucky enough to catch the TWiV t-shirt.
That's right. She's wearing the shirt
so
she caught.
This.
Look at that. So we did a live TWiV. I did a TWiV with, what's his name? I forgot his name already. The guy who writes about Nobel Prizes.
Erling Norby.
Erling Norby. And Gisa was sitting there and she caught one. Yeah, that was a lot of fun. And also from the University of Veterinary Medicine in Hanover, Steffi Becker. Welcome to TWiV.
Hey, welcome. I'm so excited being here, and I'm happy Gisa asked me to join her today.
Great. I really like when people are excited to join us because we're really excited to have you. And I just think it's great we can have people from all over the world and talk about science. And from the Bernhard Nocht Institute for Tropical Medicine, which is in Hamburg, and also from the WHO, César Munoz Fontella. Welcome to TWiV.
Thank you very much. Thanks for having me.
My pleasure, our pleasure. Now, Gisa, we were supposed to do TWiV in Germany, October 2020, right?
Exactly. Maybe I can tell a little bit, the folks a little bit about how this came about. So I had contacted Vincent in my function as one of the chairs of the German Society of Virology working group Cell Biology of Virus Infection. So the German Society of Virology is a society of around 1,000 members of German-speaking countries. And actually, everybody is more than happy to join the society. So it really bridges basic research and clinical virology. And I had contacted Vincent to host a TWiV at our workshop. So we have an annual workshop, which is for young scientists. Typically, we have 40 to 50 young scientists, young postdocs, and PhD students there. And for big shots, for keynote speakers from all over the world. And one of those was supposed to be Vincent. So that was scheduled for last year, and for obvious reasons, we had to postpone this. And that's why I'm actually happy that now we at least can meet each other. I shouldn't say virtually, right? Through this online podcast today.
I think, Kathy, you were going to go as well to the meeting in Germany, right?
Yes, I was looking forward to it as well.
Yeah. In fact,
I do have to say that I intersected with both of the places that Gisa is associated with. The small DNA tumor virus, the adenovirus meeting, different meeting, small meeting was at Umeå. And then it was also near Hanover in Barsinghausen in the sports academy there. So I know both of those places, but I'm really happy to meet Gisa today.
Yeah, that's fantastic. And we hope to be able to welcome you probably next year in Germany. So both of you, that would be fantastic.
Yeah, I was really looking forward to it. I had contacted Matthias Fischer. He's in Heidelberg, I think. And I was going to go visit him for a couple of days and do some TWiVs there. And it all, boom, gone. And I even had my plane tickets. which I had to cancel. But when I was in Sweden for that meeting, Swedish Society for Virology, I guess, many people said, "Oh, you have to visit Umeå one day. There's great virology there." So I was always intending to go back. And in fact, last summer, there was going to be a Europic meeting in Helsinki, and I was considering going back to Sweden after that, and I never did. But with Zoom, maybe there won't be many meetings anymore. I don't know. What do you guys think? Do you think you will still have in person? I know here in the US we will.
I think we will have. Everybody's looking forward to the personal meetings. So we will actually in Hanover host the annual German Society of Virology meeting in March. And unfortunately, it has to be an online meeting also this year. But I think everybody will be excited to meet again in person because in the end, all the chatting in between the sessions is what the conferences are, not only what they are there for, but what everybody's looking forward to.
Yeah. So I would like to hear a little bit about your histories first, and then maybe let's talk a little bit about the pandemic situation in Germany, and then we'll talk a little bit about each of your work. So let's start with you, Gisa. Tell us your history.
Yeah, so I'm a trained biochemist. So I studied biochemistry in southern Germany in the little town of Tübingen. That's one of the old renowned universities in Germany. And I fell in love with proteins, and that I'm still in love with them and protein interactions. Then I went on. Actually, I did my diploma thesis work in a protein crystallography lab with Wolfram Sänger. And he's one of these big shots in crystallography. He solved the photosystem 2 from cyanobacteria.
Wow.
Huge protein complex.
Yeah.
But I didn't want to stay with cyanobacteria. I was interested in infectious disease and sort of also medically relevant problems. So I moved on and for my PhD to the Max Planck Institute for Infection Biology in Berlin. And I had a wonderful mentor there, Arturo Ziglinski. And he taught me all you need to know for your scientific career, how to think critically, how to think out of the box, and how to actually also taught me the scientific writing skills, which I'm torturing my students now with. And at that time, I worked on innate immunity to bacterial infections. But I already used the technique that we're still using, and that's mass spectrometry-based proteomics to identify protein-protein interactions. And at that time, I found a co-receptor for toll-like receptor 2, one of these innate immune receptors, using proteomics techniques. And I was part of a big PhD program on infectious disease. And whenever I heard the talks about viruses, I got really intrigued and interested. And I thought, well, viruses are cool. And there's little known about viruses. So I think I want to study viruses. So then after my PhD, I decided to go and move to a virology lab. And I was lucky enough to find a position in Charlie Weiss's lab at the Rockefeller University in New York. So I went to New York for my postdoc and had a lovely time there, wonderful time. Charlie, again, was a fantastic mentor. I learned a lot. And as most of you know, maybe not some of the listeners, Charlie just received the Nobel Prize last year. He was one of the 3 laureates for the discovery and of hepatitis C virus. And so I worked on hepatitis C virus, one of these enveloped RNA viruses, like the one that's keeping us busy these days. I also again focused on protein-protein interactions at that time, on protein interactions during the entry process of hepatitis C virus. Again, used mass spectrometry-based proteomics, but I was also involved in understanding species tropism, and that is still keeping me busy right now. This question of why certain viruses infect certain species but not others. Hepatitis C is a virus that's has a very narrow tropism, only infects humans. But then during my time in New York, the first Hep C viruses in other species were discovered, actually by Ian Lipkin and others. This is very interesting. And then I moved, after my time at Rockefeller, back to Germany. Family and friends were calling. And I moved to the TWiN, which is a joint venture between the Hanover Medical School and the Helmholtz Center for Infection Research here in Hanover. and continued my hepatitis C work, but also started to work on other exciting enveloped RNA viruses. And that goes a bit back to the legacy or the history of Charlie Weiss's research. I started to work on alpha viruses, and these are very interesting zoonotic viruses. Some are re-emerging. So these are viruses that reside in animal reservoirs and that are transmitted by mosquitoes to humans and can cause either arthritis-like symptoms or encephalitis. And this is what we're doing right now, again, focusing on protein-protein interactions, mainly in the membrane of cells, so in this lipid bilayer. This is very exciting for alpha viruses because they have a very peculiar replication complex at the plasma membrane, actually. But I don't want to go too much into detail here. I guess I should just tell a bit about my career. So then after several years at the Twin Core, I recently got appointed a professor here at the veterinary school. And I moved here with my team 3 months ago under pandemic conditions, which it was not easy. I can tell you, if anybody's interested, I can tell you a lot about it, but we are very happy to be here. So Steffi is one of my dear colleagues, and this is fantastic because she has all the arthropod expertise that we don't have. So this is a great place to be. And we are located at the Research Center for Emerging Viruses and Zoonosis called RITZ, with many fantastic virologists here in Hanover.
What years were you in Charlie's lab?
I went to New York in 2009 and stayed until 2012.
Wow, 3 years. And we never met during that time, right?
We met briefly once. Juliet Morrison once introduced us at the Rockefeller Faculty Club.
Okay, right. Got it. And
very briefly.
What is the connection with Umeå University?
Yes. So, I forgot to say that. So when I came back to Germany, I applied for a position in Umeå University, and I was basically I was selected for a junior group position, and then for several reasons I could not fully move to Umeå, but they really wanted to have my expertise and have me there for research and collaboration projects. So I was very lucky that they offered me a guest professorship position, which I still have. And Umeå University is a fantastic place, very collaborative place, fantastic virologists, adenovirologists, as Cassie will know, but also virologists working on emerging viruses and zoonotic viruses. So I have several joint projects with Umeå researchers, and this is very productive.
Do you speak Swedish?
Everybody thinks I speak Swedish because I'm blonde, but so when I could still travel to Sweden at the airports, everybody would speak Swedish, but I can only say 10 words so far. I wanted to learn it, but the Swedes are so good at speaking English, so there's really no need to, unfortunately, to learn the language properly.
So Gisa, in the image behind you, obviously, you got your plasma membrane and integral membrane. What's the virus there? Do you know?
Oh, well, actually, a good friend of mine, Steve Soul from New York, he's very good at these renderings. So I made a hand-drawn cartoon and of the plasma membrane proteins, and he placed one of his viruses on top. So the membrane proteins are the entry complex for hepatitis C virus. So here we have our favorite protein, CD81, a tetraspanin, which forms homodimers, and all the other proteins that we discovered to be part of the complex. So the virus should be hepatitis C, but obviously we know that the E1E2 glycoproteins look a bit different. So, but it's not SARS coronavirus.
You didn't overlap with Matt Evans in the Rice lab, did you?
When I interviewed, Matt was still there, and of course I chatted with him. But when I arrived, he had just left.
Yeah. So Matt was, of course, at Columbia with Steve Goff, and I was on his thesis committee, so I've stayed in touch with him. So Steffi, what's your story?
My story starts at a small university in southern Germany, which is close by my parents' home. And by the age I went to university, I was not ready to leave everything yet. So I studied biology at the University of Kaiserslautern. And although I knew that viruses would be like my first love, because I knew I wanted to work on viruses when I got a book from my parents at my 18th birthday, which was called Viruses, Thieves, Murders, and Pirates. So I was fascinated by the topic, and I knew one day I want to work there. But in Kaiserslautern, there was no virology. So I did the next best and went to the bacterial genetics department and worked on Streptomyces silicolor, but not on the antibiotics, which one would think would be the most important thing working on. Nope, I worked on replication origins of those little guys. Tried to find the replication origin of a plasmid. At one point, I did mutagenesis, and obviously I found it because the plasmid vanished, and it somehow just appeared in the genome, but I never could follow up the story because then my time was over. And by that time, I managed to get to interview to the Bernhard Nocht Institute of Tropical Medicine, where César is at, actually. And I interviewed with Stefan Günther, who was, together with Christian Drosten, the first author of the first SARS coronavirus publications. So he was my PhD advisor. And I was so excited to be there. And he said, well, I do have a project about Lassa virus. Do you want to work on the BSL-4? And I was like, sure, I want, of course. Let me come here. That's my dream job. Please hire me. And he said, like, okay, we'll see if it's your dream, but I hire you anyways. So I got to work on Lassa and Ebola, which was super exciting during my PhD thesis. With Ebola, this was kind of a side project because Beate Kümmerer, she was actually also in Charlie Rice's lab. She came over to Hamburg to work there as a group leader, and she brought some cell lines with her with a ZAP, with a zinc finger antiviral protein. And she said, like, could you please test everything you have in the BSL-4 to see if it has any effect? And I was like, oh, yeah, sure, why not? Let's do that. And Lassa didn't give a shit, but Ebola was really, really strongly inhibited by the zinc finger proteins. So I got an Ebola project there. And I worked also on transcription profiles in liver cells after Lassa infection, just to find out there's not so much of transcription induction. The cells don't even seem to recognize. And I tried to develop small RNAs to inhibit Lassa virus growth, which kind of worked pretty well. It was okay, but after my time at the Bernhard Nocht Institute, I thought like, oh, I would be really interested in working on arboviruses, because I found it so fascinating of these little viruses that they are able to grow in such two really distinct environments, like a mammalian host and an insect host, which are so completely different. And lucky as I was, there was a position in Strasbourg with Jean-Luc Imler. He is in the same department as Jules Hoffmann is. So I also, during my career, met a Nobel Prize laureate, which I'm really happy to be able to meet someone like that. It's really fascinating. Jules is also a fascinating person. And I started working on Drosophila innate immunity towards viruses. And actually, I have to say I stayed with immunity in arthropods, because it was so interesting how an animal which is basically thought to be so much easier than a mammal can counteract viral infections, and how many different pathways are used. And also, to be honest, it makes experimentation so much easier. It's not this long animal protocols. I mean, César knows we together tried to get one at one point, but it was so much easier. So I decided I stay with insects. And after 4 years in France, I wanted to go back to Germany. Also, family was calling, friends were calling. And, there was a position at Bernhard Nocht Institute again. I thought like, well, I know the place. It's a great place. I love Hamburg. Let's try. But they were actually saying they are seeking for an entomologist, someone who was supposed to work on mosquitoes. So I wrote them a nice letter saying, guys, I know I'm not the person you want, but actually I can propose something else. Please take me. And again, I was lucky. And they said, yeah, okay, well, that sounds fine. Why don't you come and do your project on innate immunity in Drosophila against Rift Valley fever virus, and you work also on vector competence of German mosquitoes on West Nile, which we would like you to do. I was like, sure, I can try. And yeah, so I developed a research line working on vector competence assays, developing those assays, trying to figure out how to get enough German mosquitoes into the lab, into the BSL-free insectary, and how to get those guys take the blood meal I want them to take. Not super easy. Some of the Germans are not really bloodlusty as the tropical ones. Most of them are like, yeah, you have food? Seriously? Should I take that now? Well, I'm not so hungry, actually. So difficult, but we managed, and we did a big study to figure out if those guys are infectable with West Nile, to have predictions. That was in 2013, and we started doing our experiments, and it looked like they are very susceptible. And I was like, yes, I'm sure West Nile could establish in Germany. I'm super sure. That actually is true. We had the first West Nile cases in 2018, and now it's established virus. But I didn't really foresee where it would enter. I would bet my ass off it's entering in southern Germany. And Lake Constance would be my best guess. I was so wrong. And I'm happy I didn't bet body parts of myself, because I would have to give them to people. And in the end, West Nile came to Germany, but it used the eastern route. Anyways, it established. Now we do have West Nile in Germany. Sometimes you also wish you might have been wrong, because that's not super news, actually. But so far, it's still kind of under control. It's not overwhelming. Yeah, well, after my group leading experience, and that's where I met César first, because he was at the Heinrich Pette Institute by that time, but he was often visiting us in Bernhard Nocht Institute. And he had also a high interest in Rift Valley fever virus. So we both said, let's work together. Let's put together insect immunology and mammalian immunology to work on the very same virus from both sides. And we tried to write some animal protocols. We actually succeeded, but it was not such a big fun, I have to say. It's easier with mosquitoes. You don't have to ask authorities to work with them. You just do it. And then you just note down how many you've used. That's basically all. Yeah, and then after 3 years, I was thinking about, I should start my teaching experience. I should get my habilitation, which is in Germany usually necessary to get a professorship. But before I could actually target that issue, I saw that the Friedrich Löffler Institute was seeking for director of an institute. And a friend of mine who was there said, well, why don't apply? And I was like, are you crazy? They're not going to hire me. But then he said, well, if you don't apply, they for sure don't hire you. So I did apply, and they hired me. So I spent 1 year on the beautiful island of Reims, working together with Thomas Mettenleiter. Some of you might know, also wonderful guy. It was great. But I'm not the island guy, I realized. I'm a big city girl. I'm not an island girl. I can't live in rural places. So I was really happy when Hanover told me that they actually also want to give me a professor position. And that's how I came to the vet school in 2015.
Okay, wow, that's quite a convoluted story. Really interesting, though. I have to say, I was in Hamburg once, and there was a famous polio guy in Hamburg at the Heinrich Pett, Gebhard Koch, and he worked with a guy named Peter Nobis, who made the first monoclonals against poliovirus receptor. And I went to visit, and he let me stay in his house, which was lovely because it was this wonderful house with a lot of glass, and I stayed there a few days, and it's always nicer than staying at a hotel. And I loved Hamburg. I always said I would go back, and I never did. Oh, well. César. Yes, go ahead.
I was just going to say, it also sounds like you really made the most of opportunities and went for things that didn't quite seem within your reach, but you convinced them, like saying, well, I can do this work on insects instead of you having an entomologist, or trying for the director position. So that's really encouraging.
Yeah, that's you have to reach, right? Mm-hmm. Always. César, what is your story?
So my story is a story of luck, or finding, I think, the right people at the right time. So I studied biology in Madrid, in Spain, and I remember actually that reading the Hot Zone and Virus Hunter from C.J. Peters at that time, and I was really like attracted to viral hemorrhagic fevers, and something I had this feeling all the time that I wanted to work on viral hemorrhagic fevers. I did not do that in my PhD. I did a PhD in herpes viruses with Carmen Rivas in Madrid as well. And then, as every Spanish person that wants to do a career in science, the first thing you do is to leave your country, maybe forever. And then I went to Sinai in New York. I trained with Adolfo Garcia Sastre and with Stu Aronson, mostly on flu. But then at the end of my postdoc, I had the chance to learn to work with dendritic cells on Ralph Steinman's lab, or with Ralph Steinman's people, which actually was a really amazing opportunity. And I developed an interest in immunology. So I learned more immunology from Anna Sesma as well. And then there was a chance to go to Hamburg to start my lab. And I went to the Heinrich Pett Institute, and I had an association contract also with Bernard Noch, which is a very interesting place because it's the only tropical medicine institute in the world that has a BSL-4 lab. So you can really work on BSL-4 pathogens in the context of tropical medicine, which is really, really interesting. And then I remember this was 2011, and I remember that I was debating whether to focus on flu or viral hemorrhagic fevers, because I said, okay, I'm going to focus on viral hemorrhagic fevers because this is going to be less competitive. And three years later, we had the biggest outbreak epidemic to date in West Africa, Ebola, and then everything changed for me again. And I started to work very closely with WHO in different settings because I had the chance to participate in different outbreaks of viral hemorrhagic fevers in Africa, so very close to the R&D blueprint team. And then, I moved to BNI as a group leader in 2018. And then now I can say that I'm fortunate enough to be able to call friends of mine, people that were in those books, in Virus Hunter or in the Hot Zone. So it was a pretty interesting story. And this is what I do. So we, in the lab, we focus on immunology of viral hemorrhagic fevers. We do a lot of animal modeling, working at the interface between innate and adaptive immunity. But with years and with outbreaks, it has become also research, a little bit more operational research, if you want, or between epidemiology, public health, immunology, a lot of diagnostics, training. We do a lot of work in Africa. This is what I do and what I am now.
What's your role at the WHO?
So right now I'm seconded to the Blueprint group, and because, as you know, the R&D Blueprint has the work to identify some priority pathogens for which development of diagnostics, therapies, vaccines is a major concern. So when the COVID pandemic started, I was asked to work with them to try to set up some expert working groups. In particular, the working group that I'm chairing is on animal models for preclinical development of vaccines and therapies. So I'm very honored to chair this group with Bill Dowling and with Simon Funel from Public Health England. And then, as usual, you get involved in some other things and committees, vaccine prioritization, we are very much involved in the vaccine solidarity trial that the WHO is implementing in different parts of the world. So, trying to help in different working groups to control or to help in this situation.
I wonder if you think WHO is relieved that the US is rejoining them.
Yeah. We are all relieved. We are very happy. For obvious reasons. not only political, but also scientific. I think, obviously, I mean, I can tell you that from the people that we have in the different working groups, a vast majority of the scientists are from the US. You know? So it's a really important contribution. And I can tell you we're very, very happy, yes.
I want to talk more about hemorrhagic fevers later, but first, let's, Gisa, you said you might give us a little update of what's going on with COVID-19 in Germany. We've heard, of course, from Das Coronavirus, Christian Drosten. Let's hear from you.
Yeah, well, obviously Christian is our coronavirus expert here in Germany, but I can obviously give a quick update on the numbers. So let me, for those who are watching also the video, I can share this presentation. But no worries, I will also give an audio update. Where we are now is what you may want to call a second wave. So the virus arrived early last year in Germany, and actually we had the first wave with a peak of roughly 6,000 infections per day. So just for you to understand the numbers, Germany has around 83 million inhabitants. And they were very fast. I think the government did very well and implemented measures quite fast, like a lightened lockdown, where people were supposed to work from home office and keep the distance, implement hygiene measures, and where the mask wearing came a little bit later. But this is going very, very well now. So this first wave early last year, in spring last year, that was contained. And then over the summer, we were doing pretty well. However, after the summer break, when schools reopened and without any measures except for the general recommendation of social distancing and mask wearing, we were running into a fairly big second wave, as you can see here, already early in spring. And then the measures that were implemented came maybe a little bit too late. So we're still at very high levels now. We have today 13,000 new cases that were reported, for yesterday. So it's declining now again, but you can see that we were around 35K cases per day, which is a lot for Germany. And this was concerning, and especially now with the new variants. So where are we in terms of exact numbers? So we have 2.1 million total diagnosed cases in Germany now. And as of today, 54,000 reported deaths, which is clearly too much, right? I mean, the 7-day incidence rate, I should say, is now dropping. So 101 per 100,000 inhabitants. So we are, I think, on the right track, and I'll show later. And this is because in November, December, more lockdown measures had been implemented, and they have just been prolonged last week until mid-February, and maybe prolonged even longer. So this is how this looks by county, the 7-day incidence rate. So for those who can't see this, of course, there is variation in the different regions of Germany. Initially, last year, the very first case was reported in southern Germany, in Munich. That was an imported case. And then later on, during the pandemic, we had quite some major outbreak events in the central western part of Germany. And right now it looks more like we have higher incidence rates in eastern parts of Germany. But overall, we are everywhere around 100 in terms of incidence rate. So this is not good, because here really for successful contact tracing, we need to be below 50. That's the minimum. So I pulled this data from the Robert Koch Institute coronavirus dashboard. In terms of age group and gender, there are no big surprises in Germany. So this is what we've seen in other countries. So at the moment, we see more and more new cases also in the population in the age groups 15 to 34 and 53 to 59. So this is really, it's spreading throughout all the different age groups now. And this is obviously a concern. What we see in terms of COVID-19 related deaths is, again, no big surprise. In the 80 plus population group, we have most of the deaths. And what I'm showing here are the relative numbers per 100,000 inhabitants. This translates into total numbers of 15 to 20,000 males and females in the 80 plus group. And if you look at the relative numbers, males are proportionally at higher risk of dying from COVID-19, as has been reported. And also we have significant numbers of 5,000 females in the group 60 to 79 dying from or with COVID-19, and around 10,000 in males in this age group. So if you look at, and this is actually what the authorities are always worried about, the ICU capacities, which are fairly good in Germany, also in comparison to other European countries, but still we are running into problems with the high incidence rates. And right now we have 4,500 roughly ICU patients in Germany. And obviously we would like to get these numbers down, right? Again, you see that this follows the case number curves with the first wave early last year and the second wave now peaking in mid-January. So I thought people would also be interested in the vaccination campaign. The European Union coordinated buying the RNA vaccines from BioNTech and Moderna, and also from the adenoviral vaccine from AstraZeneca, which is not used yet or not delivered yet, I believe. César, you correct me if I'm wrong. At this point, we have 1.7 million vaccinated. That's roughly 2% of the population. And there are different priority groups based on risk of contracting and developing severe COVID-19, and also the exposure, basically the probability of being exposed. So the group 1, and that's what the German government is aiming for, to have the whole group 1 vaccinated by mid-February. Let's see if this is going to work. That group 1 comprises 6 million people. That's roughly 7% of the population. These are the ones age 80 or older, but also the intensive care unit personnel and nursing home personnel. So these are really the most vulnerable groups, and also the people at highest risk of being exposed should be vaccinated here. In group 2, that's the age 70 plus. It's also immunocompromised patients, but also patients after, well, for instance, patients after organ transplantation, but also dementia patients, mentally disabled, contact persons of pregnant women or transplant patients, and individuals in refugee homes and homeless shelters. And that's 11 million people, roughly 14% of the population. And the third priority group is people aged 60 and older, but also teachers, chronically ill, people with high BMI, all other medical personnel that was not covered in group 1, but then also state employees like police, firefighters, army, and justicial employees. After that, we have group 4, which is everybody else who wants to get the vaccine and get the shot, except for pregnant women and everybody age 16 and younger, because I guess in the clinical trials, the vaccines have not been tested in these individuals. So this is where we are at the moment. Maybe you've heard all the news about the vaccine supply. I mean, this is no difference in Germany. Obviously, there's always criticism, people saying that it's not going fast enough. But I think overall, when we look at the measures that have been taken and throughout the whole pandemic, I think Germany is doing fairly well. We have to say that early on last year, during the first wave in spring, there was a very early decision by the government to reduce contacts, to have people in home office if they can, to have implement the hygiene measures, mask wearing, etc. And so we could really control this first wave fairly well. so that during the summer also restaurants opened again and bars could open again. But then now, as I said, since last fall, we are running into this second wave. So we are still in a lockdown right now. What the government has done in December is to provide FFP2 masks for those age 60 and older. So you can get those in the pharmacy for free. And now what's new since early this year is that in some states in Germany, you have to wear FFP2 masks on public transportation and in certain settings indoors. In other states, you can also wear just surgical masks, but but there's really you would get fined if you wouldn't wear masks, and because we've seen that they work. And vaccination campaign started around the change of last year at the end of December. So this is where we are in Germany right now. We are expecting that the situation will continue to be difficult until spring. And now with the new variants, so all the 3 new variants have been found already in Germany. We are increasing also the sequencing efforts. They are not as great as in the UK, for instance, but the capacities for sequencing are increasing now. So we hope to have also these new variants under control, because as many of you know, there's now good evidence that at least some of the variants may escape the antibody responses and the immune responses elicited by the vaccine. So we need to be aware of this. And César, definitely feel free if you want to add anything.
Yeah, I was going to say before you move on, just for the listeners who might not know the German, the cases are on the y-axis, and the black curve is deaths, the aqua curve is recovered, and the blue curve is active cases. And then everything else has pictures by it, so you can probably figure that out.
Yeah, I can also explain more, but I thought,
I think it's fine.
Exactly. Yeah. Thank you, Cassie. Yeah, it's true. So for those seeing the graph, exactly as Cassie stated, so the active cases are the light blue ones. Yeah.
So BioNTech is a company in Germany, right?
Yes, that's a company based in the center of Germany, and they teamed up with Pfizer to basically now distribute the vaccine and produce the vaccine. 2 companies in Germany that have developed RNA vaccines that initially focused on RNA vaccines for cancer therapy and other diseases, and during the pandemic have used the technology to make the SARS coronavirus 2 vaccines. The second company is called CureVac. That's from southern Germany, from Tübingen. Yeah.
So I remember we talked to, we had Christian on in September, and it was striking that at that time, Germany was not having much of a problem. I recall feeling almost envious or chagrined that we were having as much of a problem as we were having when Germany seemed to be in pretty good shape. I'm interested in your perspective on sort of what happened that resulted in this second wave.
Yeah, I mean, César and Steffi, you're welcome to also join in here. I think, we had, of course, the schools opening after the summer break. This could have contributed. I think people also may have gotten a little bit less compliant when it comes to the contact reduction measures. I also know that in the first wave, a lot of employers allowed people to work from home office or recommended this. And this sort of people, I think, got a little bit too relaxed about the situation over the summer. So maybe people did not pay enough attention. I think the wave that we see starting last fall cannot yet be contributed or attributed to the new variants, I think, but one would probably need to go back and sequence some of those samples. But I think it's really human behavior that caused this second wave.
Yeah. So this is the lesson over and over again, is that the burden of virus in a given population seems to be a direct consequence of whatever sort of behavior people are undertaking. It's amazing.
Absolutely.
Amazing.
So we should learn our lesson and be careful now with the new variants that may spread faster. Yeah, I know. But I think it's important to tell everybody. And I mean, COVID-19 also for the younger generation is not a disease that you want to catch, right? I mean, we know of severe cases also in the younger generation. We know that people can have long-lasting consequences of the disease. Of various,
Nevertheless, one of the things about your presentation, you had two slides back to back. One was where the cases were, and the other was where the deaths were. And once again, as in the past, I'm struck by the difference in those distributions, that the cases are in the younger individuals and the deaths are in the older individuals. And to me, this is one of the most insidious things about the disease, is that with the lower mortality and the people who are in the younger people, they're probably interacting in a fashion that promotes the disease in those populations. And, you know, it doesn't mean as much to them. As you say, people do get sick and die in those, but it's not as much of a problem. But as a consequence, it supports a larger burden of disease in the population, and the older people get trashed.
Yes,
okay?
You're absolutely right. So it's a call for solidarity. And there was a big call early last year during the first wave in Germany. And I remember that all the people, everybody that I know from the younger generation, they all said, well, we have to do this. We have to stick to the rules. We have to protect the vulnerable, the ones with preconditions, the elderly. And I think now this solidarity may be a little bit less pronounced, let's put it this way.
And solidarity is a great word for it. I really like that. And it's not, although it is to some extent a political solidarity in this country and as well, but more importantly, it's a generational solidarity. Yes. Okay. That's really important.
It is. Yeah,
Steffi, sorry, go ahead.
I just had one other thing. In this country, we've had difficulties with vaccine distribution. Okay, and I'm wondering whether the healthcare situation in Germany is such that it mitigates that problem. Whether you've had problems with distribution as well.
Yes, I think every country is facing these problems. So in Germany, it's also all the vaccines are bought by the federal government and then distributed to the states, and the states have to set up the vaccination centers and distribute the vaccines. I know in some states it was difficult, or especially for the elderly population, you had to sign up to get vaccinated online, which is not for 80 plus for some not a good option. I know that there were different strategies of holding back the second dose or not holding back the second dose, because you've all heard maybe that the BioNTech vaccine, they are having some issues with the production. Well, not issues, but they are scaling up the production. But in order to do that, they need to halt the production for a while. So there are issues, but I would say, and César and Steffi, you can definitely comment. I think overall it's going quite well. Everybody was hoping it would go much faster, but we're in the middle of a pandemic. I mean, we all have to remember this, right?
Yes, Cesar, go ahead.
Yeah, this is actually, I think it's an interesting lesson how the rich countries, we really suck at vaccinating people. Like, we're really bad. I mean, I can tell you that in Guinea, we vaccinated 250,000 people in a week, because the change of opinion is much less. I mean, you have a leading institute, like a leading body like WHO talking with the Ministry of Health of a particular country, and then everything is done, and you talk with the necessary people in the cities, in the villages, and then you get the job done, But in Europe, you have to talk with the government X that then needs to talk with the sub-government whatever, and then the Ministry of, and it's a logistic chaos. We are like one spoiled kid, playing video games that all of a sudden needs to run 100 meters. It is incapable of doing that. And what is sad of this situation, I think, is the fact that now all the rich countries made all the bilateral agreements with company X and Y, bought all the doses. There is a lot of vaccine hesitancy in the population. So you have all the vaccines, but people don't want them. A lot of people don't want them. You don't know how to logistically do it. And then you have the rest of the countries that don't have access to the vaccines. I think it's crazy. It's really sad.
Cesar, do you have any involvement in the WHO's effort to ensure equitable distribution of vaccines?
So I'm working as a consultant for the vaccine solidarity trial, but just for the immunology part. But I'm also part of COVAX, which is this facility that's working for equal distribution of vaccines. So I know of some of the struggles, and it's something that really worries me, because obviously I think something that we all have to understand is very clear that you need to control this pandemic globally, because unless you do that, you're not going to be able to trade with another countries or travel, and it's really worrisome. And I really don't know how are we going to do it, but I think it's important to remember that this needs to be controlled at the global level, not only in a particular country.
César, has vaccine been distributed to, say, African countries in any numbers?
Not that I know.
Wow.
But I don't quote me on this, because I may be wrong, but not that I know.
Yeah. Okay. Steffi, I wanted to talk a little bit about schools. So you're teaching, and have schools always been open in Germany throughout the pandemic, or have there been lockdowns on schools? Tell us what's up.
Well, through the first wave, when the government decided the first lockdown, we had a complete lockdown. It was just start of summer term. And we had absolutely no presence lectures. Everything had to be switched online. And this was really a big hassle, because I think the decision was made March 19th. And in April, I think 10th or 12th, there was start of semester. So we were supposed to teach. And we had practical courses, of course, to lead. So, we were stuck okay, how supposed to do that? So it was a big thing. And how to do, should it be Zoom online lectures, or should we just record our lectures? That's what was actually, the decision was made to record all lectures, which is not so nice, because you really, for me, my feeling, you don't even see somebody you're talking to your computer. And then still you're afraid, because it's recorded. If you kind of use the wrong word, or if you're just messing up a sentence, it's there forever, and the students will listen to it, and maybe they will make a drinking game out of you. You don't know. So you're kind of, okay, I have to do this slide again and again and again. And after the 10th time doing the same slide, so tired. So yes, it was really hard. My first thought when I heard it's online, because in summer term I have to teach quite a hell of lectures. So I was like, oh, that's good. I have more time for papers and grants. And then I realized how much work recording lectures is. But by that time, I had already started the process of writing a couple of grants and papers. And I was like, I have to finish all of this at one point. So first summer term was hard. And then we had the big discussion, because we are a veterinary school. And, you all know you can't be a veterinarian without working practically with an animal. So at least we had to do some hands-on teaching. It's impossible. And even for the master of biology, what kind of biologist would that be who had just done his master studies for 4 semesters online? It's difficult. Well, of course, if you do bioinformatics, everything is cool, but the other disciplines really difficult. So we started in summer to discuss how to have a hybrid semester, which means we reduce the practical work to the necessary minimum to ensure that this class of veterinarians will be still able to treat an animal. But we reduced contacts, and there were plans made that people should be tested, and that they are given the cohorts are smaller than they usually are, and they are fixed, because sometimes, usually they start switching when they have kind of times which are not good for their a side job, or we also have people that are already parents. So if they have a lecture late in the evening, that's not really good. And so they interchange, and that was forbidden then. So they had to stay with their cohort because each cohort was treated as one epidemiologic unit. So if one case turns up in this cohort, everybody has to get tested. Yeah, well, and then we also try to push our lectures because we hope it's getting better. It will get better, and it didn't, unfortunately. So we started in January with our practical units for microscopy. We are teaching parasitology. I'm a virologist, but I'm teaching parasitology. Things happen. It's because of the arthropods in my life. So I ended up in veterinary parasitology, and I'm teaching entomology and protozoology there. And well, yeah, we had to show them how to use a microscope and how to identify a parasite. Now we have even smaller groups. So within a lecture hall that usually would hold 140 students, we have 40 maximum. We have put green dots so they can see where they're allowed to sit and where they're not allowed to sit. And yeah, it's way different. And I have to, of course, give the same course more often in regular terms because we have 280 around. So we have 2 cohorts. I have to give the same lecture twice. Now I give the same lecture 6 times, which is at the last time, it's really hard because I have been saying this so often. I want to stop. But of course, this cohort has to hear the information too. On the good side is, I think it's very efficient for the students because we have taken everything out of these practical lectures, what we can. So it's just showing them how to learn how to use the microscope, how to identify a parasite and what's the most important points. So it's really condensed. And because it's a lot of space in between the students, they are not allowed to interact. It's very calm. Usually when I start the lecture, I really have to scream in the microphone to make sure that they hear me because, of course, they come in, they start chatting, and it's a bit loud, and it's 140, so it's super loud. Now it's less, and because of the large distance, it's very quiet. So for me as a teacher, partly it's easier. They are all super focused. They are all very quiet, which helps me to get through with the lecture. On the other side, it's also kind of, they are losing a bit of the normal student life, which I really feel a pity for them because student life should be also interaction and chatting with your colleagues and all of this. And this was what they can't do. Well, but we do our best. And for the biology students, we have them now on single student base in our labs. So we have to do more practical classes because we take them one by one. Because also there's restriction how many people should be in the lab. We have rules how many people can be in one room, depending on the room size and on the air exchange. And we have lists, we have Excel sheets where all my PhD students have to sign in the beginning of the week for the whole week to plan their experiments. So there's never overcrowding of the labs. And students from the master studies have to be fit in this. So it's difficult, but I think we manage okay. So far, students are okay. They most of them did not really object to the whole procedure. Actually, I get a lot of emails that they're really happy that we at least try to give them some practical experience. Yeah.
So
that's how it is.
This idea of having to give a lecture, many times it makes you have greater respect for actors and musicians who perform every day. I never knew how they could do that, right?
No idea. It's so hard. Because you think like, okay, did I tell this cohort? Did I say that already? Didn't I? You feel like you're developing some degenerative disease because you're not sure. Did I say that? Didn't I? Yeah. Well, I just repeat, and then the students think you are suffering from some neurodegenerative because you repeat yourself all the time.
I always have the thought running through my head, even if it's just something that I taught last year and now I'm teaching it this year. don't they know this already? I've already said this before.
Yeah,
So it's
right.
kind of like, a frustration. And I can't imagine having to do it 6 times within a short time span.
Yeah.
It just would, really try my patience, I think. So hats off to you for that.
Gisa, have you had are you able to continue your research, or did you have to be diverted to some SARS-CoV-2 in your lab?
Yeah, so actually when I was still at the Hanover Medical School last year during the first lockdown period, sort of, let's call it first wave, we were working in shifts actually to reduce the number of people in the labs and in particular in the offices, because the students' postdoc offices are typically packed. Well, actually, maybe US researchers don't know. In US, you have this nice setup where you have your desk next to the bench. In Germany, you have lab space and then you have separate office space, with the advantage that you're really allowed to eat and drink there in the office space. And those spaces are typically packed. So we had to work in shifts, actually. And at some point, it was only people working on SARS coronavirus 2 allowed to work in the labs, at least at that. But that differed from institution to institution. I think here at the veterinary school, it wasn't that strict. No. But we did get involved fairly early on in also SARS-CoV-2 work, because, we are RNA virologists. We know our envelope viruses. So we established the systems, VSV pseudoparticle systems, to study the entry of the virus. But also in the BSL-3, because a lot of my team members have BSL-3 experience from the HCV and the chikungunya work. Established the isolate that we got from Christian Drosten in the lab. And of course, a lot of clinicians asked us to help out. So we were involved with Thomas Pietschmann in one of the Bill and Melinda Gates funded repurposing screens for drug repurposing. We also helped clinicians who tested sera from convalescent patients, whether those could neutralize the virus and could be used as therapeutics, because at that time we didn't have any Regeneron antibody products or anything. So we got involved, and that also allowed my students and postdocs to still do work and to do meaningful work. And I think everybody was happy for that. Now, then over the summer, everything was relaxed. Now here at the veterinary school, we are actually still allowed to do lab research also on the other viruses. But everybody, as I said, now the rules are getting stricter in terms of everybody doing administrative work. So you really should do this from home office, which I think makes sense. So if there's no necessity to come to work, to commute, or to get in touch with other people, to have contacts, then you should not come to the university and stay home. And all the lectures, as Steffi said, are given online. So we speak to this black wall. But it works fairly well. And also there were no major outbreaks here at the university. So I think we managed quite well to keep everybody safe.
I've gotten really used to the online lectures, and now I kind of like it because students are more vocal. They're more likely to ask questions online, and they can type in the chat. And I often notice they're making comments to each other, not meant for me. They're not questions. They're making comments, which are really, and I often get distracted because they're funny, right? They're like chatting with each other, which in a lecture hall, as Steffi said, it's annoying, right? But kind of neat. I mean, I do want to be back in front, but I think it's quite interesting.
Yeah, it does have pros and cons. It's true. I mean, I think in the smaller seminars where the students know each other already quite well, and it's a good format. I mean, and honestly, I'm happy we have these formats, right? I mean, just imagine this pandemic would have happened 30 years back. When I did my first student exchange in Montana, I forgot to tell you about that. I was at Montana State University in Bozeman for a semester. And at that time, we didn't have any Skype or anything. No Zoom. The newest thing at that time was ICQ, this instant messenger. I don't know if anybody remembers that. Yeah. Cool.
Yeah. Steffi, has your laboratory research continued, or was it distracted in some way?
No, actually, I was really lucky we could continue, although, as I said, we had to reduce people in the lab. So we had to better coordinate who's working when in which lab. But also I'm in the lucky position. I have two different labs. So I have one at the research center where Gisa is too, which is pretty packed because there are a lot of people. But I do also have some labs in the parasitology, and we do have a lot of climate chambers for our little mosquito friends. And each of these ones can be seen as a single unit. So each one, it's 5 of them a person could work in if the person is alone. So we had some space we could kind of share. And the veterinary school started to get involved in coronavirus research, but they didn't force us to shift. And we didn't. We stick with our research on Rift Valley fever virus and also on tick-borne encephalitis virus, which is one of the most important causes of encephalitis in Central Europe. And actually, one of the guys that kind of profited from the first lockdown of SARS coronavirus 2, because when the government decided to have a lockdown, and to close all bars and clubs and restaurants, German people went to the forests. I don't know if American people did the same, but our people went all to the forest because you can't be locked in your house for a whole day. You go crazy. So people went out to the forest, and that's where the ticks were waiting for them, happily, already, clapping their tazi, like, yeah, there's more guests. And actually, we see that. I've just looked it up today, and there is 706 TWiV cases last year in Germany, and this is nearly double of the normal case rates. So this is exceeding even the record year of 2018, which was one of the hottest summers we ever had, and we thought that's already the record of TWiV cases we can have. And this year, we just exceeded that by more than 200 cases. So it's amazing. Yeah. And I also can say there were a lot of ticks. So in March, when they started discussing lockdowns, I told my students, all of you out to the forest, collect as many ticks as you can. I'm not sure if we can go out later on to collect them. So we do all vector competence studies now. Everybody does that. And so I send out a armada of students to collect ticks, of course, all vaccinated against TWiV. That's a prerequisite for them to do the job. And we had 500 ticks, I think, collected in one day in Hanover for our infection studies, which was amazing. Helped us a lot. And the whole year, then they didn't lock us in totally, so we could continue working on TWiV. And it was, for TWiV research, pretty good year, I have to say, from my side. For some of my partners from our TB Nature Network, which is our TWiV research network in Germany, it was not as good. Our coordinator, Gerald Doppler, for example, I don't know if anybody knows him. He's our German expert on TWiV, and he works with the German army. And of course, the army directed all forces towards coronavirus research, which is totally okay, but it took some resources from the network. So it was a few of us, and luckily me as well, to be able to work on TWiV and to keep on working on the network. I mean, of course, if anybody knows Gerhard, he wouldn't be forbidden to go collecting ticks. He loves them so much, and he's so much really living for his research. So, of course, he went every weekend collecting. He and his wife went every weekend collecting ticks, and they worked like crazy on the weekends and all their free time to keep the research going. But, of course, it kind of was a bit difficult because work, was supposed to work on coronaviruses.
How does one collect ticks? I usually try not to collect ticks.
Well, there's different methods. What we do for our infection studies, we are looking for host-seeking ticks, so the ones that have not fed yet. And this is actually super easy. You take a white sheet, so maybe a bed sheet, and you walk through the forest, dragging this bed sheet behind you. Your person tells the tick, ooh, yummy host, because you're smelling of yummy host. And it tries to grab the next thing that smells like yummy host, and that's the bed sheet. It doesn't have to be white. Ticks don't care about white, but it's easier to find them on a white bed sheet. It looks like a bit crazy when a lot of people walk around the forest with dragging a white bed sheet behind them. But that's the most efficient way collecting ticks. If you are interested in ticks that already have been on a blood host, we have been working together with hunting societies in Germany. So when they have these big social hunts where they have 20 hunters and they are after a certain kind of animal, like maybe a deer or wild boar, they shoot a lot of animals that day, and we ask them to be there before the animal goes to the cooling chamber to collect all the ticks from them. So we were like in the row, there's the hunter bringing the corpse and somebody is hanging the corpse in the cooling room, and then my students like collecting all the ticks from them, and then it goes away for the cooling and to be sold later on. Was pretty nice. We were received by most of those hunters also very nicely. They found us funny, actually, I think, because most hunters in Germany are still male, old men, and my students are mostly young women, because biologists is, I think, 80% women. So it was like a lot of young, small, tiny women standing there and collecting the ticks from the dead game animals. Yeah, it was a great experience, actually.
Rich, you remember in Galveston, we visited Dennis Benty, who collects ticks, and he had some ticks, and maybe you know this, Steffi, that actually chase you. Yeah, you don't have to get the sheet. You stand there, and they run up, and you can have them come up your hand and it's amazing. And he sent us a video of that. Yeah. Cesar, have you continued with your laboratory research, or have you been diverted to COVID-19?
No. Well, I was diverted myself, but regarding I actually decided I didn't want to do direct research because didn't want to go into potential conflicts of interest with funding and so on. So we keep working on viral hemorrhagic fevers, mostly filoviruses. I mean, in a way, we had to keep the work going because of the maintenance of BSL-3 and BSL-4 lab. So at some point we had two teams. So one was like the active team, and the other team was at home, in case someone got sick, and then they could replace this team. But the BSL-4 experiments are still ongoing. What we lost is a very important part of our research, which is the field research in Africa, because we used to spend a number of weeks per year in Africa. In collaboration with different partners and different projects, and that we lost completely, and it's really bad and really unfortunate, because it takes a long time to build those relationships, and then maybe some people change politically, so the next person in the Ministry of Health doesn't really know who you are, and it's really complicated.
What was the goal of that? What was the goal of those field studies?
Well, we do different things, but so we have programs like training programs. We have a summer school in Brazzaville in Congo that we couldn't do. So this is like an educational project entirely. We have been involved in diagnostic training, for example, through the German Foreign Office in cooperation with specific countries. And then what we have is some epidemiological surveillance projects in collaboration with Vincent, actually, from NIH in Congo, where we are looking at cohort. I mean, Vincent is looking at the wildlife and looking at what different viruses are there, and we are looking at the human component. So we are looking a lot at the interaction between humans and wildlife. For example, we are following a cohort of people that are in close contact with great apes, because they work at the national parks in Congo. So we've seen that some of them have antibodies for Ebola, and we want to understand from the standpoint of immunity what does that mean. So try to collect more information about whether, what kind of immunity they have. So all this is like different projects that we do. But we had some project with Ebola survivors in Guinea, trying to understand the duration of immunity, things like persistence. And these are usually multi-institutional projects. So, we're involved with other people. And this we lost. And then, so let's see how we recover from that now.
Is there currently any Ebola activity in Africa?
Well, I mean, as you know, we have just had an Ebola outbreak in DRC. It looks like since 2014, we have Ebola outbreaks every year. I suspect that this is because there's much more surveillance going on, so people are looking more into what is going on. So right now, there are no cases, but I suspect that because of this increased surveillance, we're going to see more and more outbreaks. Yeah.
Is the vaccine widely used, or is it only used when there is an outbreak?
It's only used when there is an outbreak. So the vaccine was used through emergency use authorization during the outbreak in DRC and also in Guinea. For example, there was also WHO vaccination campaign for healthcare workers at the border countries with DRC, Uganda, Rwanda, Burundi. But it's not. It's only used during outbreaks.
And I guess that's logistic and economic reasons for that, right?
I guess so. I mean, in reality, it would be very difficult to start. I mean, where should you start? No? Because you cannot really predict where the next outbreak is going to happen. But obviously, it has a lot of economic implications. I mean, for the same reasons that we don't have a Lassa vaccine yet. Still, Lassa is killing thousands of people every year in West Africa. So that's probably more because of the economic implications, I guess.
And when
you have
just one more. And when there is an outbreak and the vaccine is deployed, it seems to be work, right? It's highly efficacious. Is that correct? Yeah.
Yeah.
Okay.
Yeah.
Go ahead, Rich. Sorry.
That was my question because this is if I've got my history right, this is really the only virus vectored vaccine that's been deployed widely in humans. And so our experience with that in terms of its efficacy is good background for what's coming online, although it's different vectors and stuff. Nevertheless, good experience for what's coming online for coronaviruses, some of those being virus vectored.
Yeah, and it's actually a little bit the opposite story to corona, right? Because this vaccine had been there in the drawer waiting to be used for 10, 15 years. I think Heinz, Feldman, and others did this vaccine many years ago, but there was not really a market, if you want, for this vaccine. There was not really, it was not really interesting, no? But now you can see that it really is.
And I think it's really interesting that it's a VSV vectored Ebola spike, right? And the VSV vectored corona spike was just canned by Merck. Didn't work in a phase one. Yeah. It's very interesting. So you can't assume it's going to work for everything you try.
Now, the other thing that occurs to me, of course, I don't know what the Merck data actually looked like, but the mRNA vaccines and the vectored vaccines that we've seen so far, but in particular the mRNA vaccines, have set a very high bar for efficacy.
Indeed. They're getting we get spoiled, right? Essentially. Yeah. Now, Cesar, you have some experience with influenza from your years at Mount Sinai. So has there been an effect on flu in Germany during the pandemic? Do you know?
So there was a big spike of excess mortality of flu in 2018 in Germany. There was a very bad flu year. But I think that I saw a graph the other day, and I'm sure Gisa and Steffi may know this better than me, but I think it's gone. It's completely gone. Like you see the peak of the seasonal flu started in the winter, and then it's really gone. So, and this actually we had a discussion the other day with other people about the reasons, no? But why would a mask annihilate completely flu but not COVID? And I think that one possible explanation would be that COVID depends much more on super spreading events than flu. So maybe it's less. Yeah, so you need only a few super spreading events to keep the thing going. Maybe with flu but it's really interesting actually.
Yeah, we've
yes, indeed it is. A friend of mine works in a diagnostic lab in Munich, and he already told me in summer that in the spring wave of COVID, at that day when they started the mask obligation, that the flu cases were just cut off. They didn't get any flu cases anymore. At the moment when the masks came up, nothing.
Huh.
Yeah, so there's actually...
It seems that masks pretty much contain flu.
We actually see a drop not only in the respiratory viruses, but also in the enteric viruses.
Huh.
So I have a research consortium on norovirus. That's a little bit of the odd pet in our collection. So we also work on gastrointestinal diseases. And we wanted to start a prospective study early last year. No more reported norovirus cases in Germany whatsoever, or well, very few. And if you look at the statistics for also rota, other enteric viruses, I think the hygiene measures really help. So of course, we cannot say whether it's really the hygiene measures, people wash their hands more often, or the social distancing, or whether people just don't go and see the doctor anymore as frequently as they would have done before. But the notion is really because if you look at bacterial gastroenteritis, the case numbers don't drop that drastically, because some of them may be foodborne. So I think it's really some impact of mask wearing for the respiratory viruses and hygiene measures. They do help.
That's very interesting, because with the GI viruses that are basically fecal oral, the aspect of the hygiene measures is different. You wouldn't expect the masks to really affect that, except that you're not putting your hands in your mouth as frequently. But it's a different aspect of the hygiene measures that impacts those compared to the respiratory viruses. That's interesting.
To put together some data now to see whether this really, but it really looks like it coincides with the respective measure and when they are implemented in different countries. Yeah.
So you mentioned respiratory viruses. Aside from influenza, does Germany track any other respiratory virus infection, and have they been changed as well? Do you know?
That's a good question. I'm wondering about the RSV data.
RSV, adenoviruses, rhinoviruses, even common cold coronas, right?
Yeah, absolutely. I don't know from the top of my head. I think there's data there for RSV and rhino, but I don't want to state anything wrong here. Yeah.
I think the TBE story is fascinating, that it went up because of a change in human behavior, right? That's fascinating.
I do think that I heard, but might be not solid, that all infectious diseases have dropping case numbers in 2020, except TWiV. And of course, coronavirus.
All right. I think we're reaching an hour and a half. I don't want to take much more of your time. Is there anything else anyone wants to say before we say goodbye?
Very quickly. I want to go back to something that came up early, and that is West Nile showing up in Germany. I'm curious as to, in the U.S., this showed up very at one focal point in New York in the east, and then over a period of 2 years swept westward across the country and killed 90% of the crows, and caused an issue in the humans, and then has sort of, I guess, settled down to a new equilibrium. I'm wondering about in Germany. Obviously, the virus doesn't care about international borders. If you have any sense for where it came from and how it's manifesting so far.
It's a lineage too. So, and it looks like it came from the eastern road via Hungary and Austria. Austria has experienced cases long before us. And it showed up first very focally in some wild animal parks in Bavaria and in, I think it was Saxony and Saxony-Anhalt, so the eastern parts of Germany. That were the first cases, and the cases were seen because it was gray owls and some other predatory birds which show really strong disease. So those guys get really sick, and then they have been diagnosed. Whereas regular German blackbirds and other birds do not react as severely as the American crow does. So it's not as obvious. But from what I know is that it's since 2018, it's found every year, and it's found every year in more places. But it's not the big wave that has been seen in the US. It's more like little dotties, and more dotties are popping up. It's not like whoosh, going over the country, more like little small dots that start to show up at more and more places.
So, Rich,
interesting that it shows up starting off in zoos, same as
New York,
in New York.
So, Rich, would you say in the U.S. that was a murder of crows? Steffi, what's going on with Schmallenberg virus in Germany?
At the moment, not so much to my knowledge. So it's not overwhelming currently, but regarding animal diseases, African swine fever is coming up. So that was what we were afraid of for years, and we kind of saw it coming, which didn't help us. It's showing up now, and it raises big concerns, of course, because you might know that Germans do like eating pork meat quite a bit. That African swine fever is like Ebola for them. It kills them very rapidly. And so Germany is very much afraid, because this would be an economic catastrophe. If it would show up here in some swine producing company, it would be horrible. So they try to, but I'm not sure if they manage.
And that's we discussed this some time ago on TWiV. That's a huge problem, because it hangs out in the wild boars without necessarily causing serious disease in them. Okay, and can be transmitted. You would get in. Let me see. No, in boars, it's probably direct contact that's transmitted. In the natural host in Africa, it's tick-borne. Mm-hmm. Okay, but I think in the boars, in a situation like Germany, it's the transmission is not clear, but I don't think it's tick-borne. Is that correct?
That's correct. We do not have the tick species that is transmitting it in Africa in Germany. So far, as to my knowledge, it has never been found in hard ticks, which are present in Germany. And the African tick species so far has not been found in Germany, which is a good thing, but there seems to be direct contact transmission. And yes, wild boar is a big problem. That's why German hunting societies have started to raise the numbers for boar that should be shot, because they thought to reduce population would reduce our risk. But it's not really clear if these measures of hunting more boar really had to reduce population, because it looks like they're compensating that very well, and population sizes are just rising.
Yeah, what comes to my mind is that ultimately you have to control it in boars. And what comes to my mind is rabies. Okay, because the way that has been dealt with, being, having a wild animal reservoir, is a vaccine that's dropped in bait. And so that seems to me would be a reasonable goal, except, as I understand it, there's no vaccine for African swine fever, right?
Yeah, not as far as I know.
Despite great efforts on the part of many different labs.
Yeah. Not as far as I know. I've not heard of from my colleagues at FLI, which are dealing with African swine fever. I don't think there is a vaccine.
Do we know if SARS-CoV-2 has infected non-human animals in Germany, as we've seen in other countries, mink and cats, dogs, etc.?
So far I know, but I think Gisa knows better. There have been cats that have been infected and that have been also infected quite a while without being severely sick.
Yeah, so here at the veterinary school, some colleagues have identified cats that were SARS-CoV-2 positive. But we don't know whether they are really also transmitting back to humans. So there are no studies so far in households where people have positive cats, whether there was a sort of a spillback. So far the risk is rated fairly low that humans would get infected from infected cats, so that you would have transmission from the pets to humans. But the cats can be infected, yes. Mm-hmm.
Cesar, you mentioned non-human primates in Africa and contact with humans. Seems like they could get infected with SARS-CoV-2 there, right?
Yeah, that's a good point. Definitely, that could be a possibility. I don't know how frequent are the interactions between apes, great apes, and humans. At least, I mean, some of the, for example, in national parks, there's some groups that are accustomed to tourists and rangers, and so yeah. I mean, I think that I was just listening to Gisa, and I remember that there's also a study now from Chile showing that actually cats could actually act as reverse zoonosis mechanisms to actually pass the virus to their owners in the household. So I think that there's probably a number of animals that can maintain this virus in nature, and I think it's going to be very difficult, even if we control the pandemic, to really eradicate, let's say, this virus. I think it's almost mission impossible at this point.
Yeah. Yeah, but if you have non-human primate samples that you're looking for Ebola, you could look for SARS-CoV-2. Would be very interesting.
Yeah, absolutely.
Right. All right, let me ask you one more question. Gisa, if you had not been a scientist, what would you have done?
Yeah, I am actually, one of my hobbies is flying. So I like paragliding, or hang gliding, I think is the correct English term. Cool. And I like to be up in the air. So I'd like to be an astronaut in my next life and do virology experiments in space if I could.
we know someone who does that, right? How about you, Steffi?
Well, when I was younger, I wanted to be an artist because I would spend all my afternoons drawing. So, but at one point I realized that I might not get paid to make a living out of it.
Yeah, that's a tough one. How about you, César?
So I wanted to be a tennis player. So I guess I'm not good enough, to be a tennis player, but I would have liked probably to be involved in tennis as a coach or working in the tennis world. I may think about that still.
That is pretty good, I should say. Cesar, you're pretty good playing tennis.
How do you know?
I was told.
Oh, okay. So maybe good enough at the scientist level.
All right. That is TWiV 712. You can find the show notes at microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And if you like what we do, consider supporting us. We have many new supporters. We appreciate your help in keeping this going. You can go to microbe.tv/contribute. Our guest today from Hanover, Germany, Gisa, Gerald, thank you so much for joining us.
Thank you very much for having me. And Kathy and Vincent will be happy to have you here in Germany next year, hopefully.
Thank you.
That's 2022, right? Mm-hmm. I think we can do that. Yeah, I think we can do 2022 for sure. Also from Hanover, Germany, Steffi Becker, thank you so much.
Thank you so much for having me. It was great chatting with you. And I support what Gisa said. We would be happy to have you here in Germany. And we would love to show you our country and discuss and chat and have you visiting our labs. and I show you how to catch ticks in the forest if you like.
All right. I've been to Germany more times than any other country, mostly because of meetings and so forth. I really like it. And the last time I was there, Rich and I went, and Nels Aldi. It was great. We had a blast. Great fun. By the way, the meeting was going to be in an abbey last time, right?
Exactly. So this is a beautiful monastery in the forest, and we have the meeting typically in fall. So it's really nice foliage. So you have like the Indian summer, and it's a beautiful place. And there's always a wine tasting in the cellar one evening. So you should definitely come.
So it's at the same place every time. Yeah.
It will be at the same place. So
Lovely.
I'm not hosting the workshop anymore. So that's now next generation. But and also actually, if I can say this one sentence, I can recommend all the young scientists to join the workshop. And there will be great guests next year, as you just heard.
Do you have a website for the society?
Yes, I can share the website maybe in the show notes.
Okay, that would be great.
Yeah.
Wonderful. And also from Hamburg and the WHO, César Muñoz Fontella. Thank you so much for joining us.
Thank you very much for having me. It was a pleasure. If you ever want to think about doing a TWiV in Africa, you can count on me as well.
Oh, that would be great. In fact, Daniel Griffin, our clinical host on TWiV, is often talked about doing that because he has connections. Of course, he hasn't been there in a while, but that would be a blast. I can imagine it would be a lot of fun. And since you talked about tennis, Steffi, when I see your name there on the Zoom, I think of Steffi Graf.
Mm-hmm.
Unfortunately, I'm really not sporty at all. I do have problems, coordinating my limbs. I'm a really super bad tennis player.
It's okay. It's just that the name remind me. Oh, let me ask Cesar, who was the greatest Spanish tennis player ever?
Well, obviously, it's not only the best Spanish tennis player ever, it's the best tennis player ever, Rafael Nadal.
Yeah, I just wanted to hear you say that, yeah. Okay, joining me today from the University of Michigan in Ann Arbor, Kathy Spindler. Thank you, Kathy.
Thanks. This is a lot of fun, and it was really fun to meet and see Gisa, Steffi, and Cesar. Thank you.
She says it right. She says Steffi, which, I say just Steffi. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time, and great to meet all of you. It's a delightful conversation.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.